Welcome to the Deep Dive. We're here to cut through the noise and get you well-informed fast.
That's the goal.
Today, we're zeroing in on something that's, well, quietly reshaping healthcare, the potential of wearables and smart sensors. And we're going way beyond just counting steps here. We're talking about how these devices, maybe like the one you're wearing right now, could actually transform how we develop life-changing medications.
That's absolutely right. Our focus today is specifically on digital endpoints, or DEs, as they're called.
DEs.
And we're going to unpack this, well, really complex but totally vital challenge, figuring out what a meaningful change in the data from these devices actually means when we're developing new drugs.
Right. What does better look like in this flood of data?
Exactly. So think of this deep dive as your shortcut to understanding this really cutting-edge area. It's something that's poised to transform clinical trials, patient care, really right now as we speak.
Okay, so our mission here is to pull out the key insights from a really important new paper. It's titled Digital Endpoints, Establishing Meaningful Change Thresholds.
That's the one, by McCarthy and colleagues, just published this May 2025 in Therapeutic Innovation and Regulatory Science.
Got it. And we want to help you understand the nuances of how this continuous passive data, the stuff from your smartwatch or similar tech, is really changing the game in clinical trials.
And the core question, the thing this paper really tackles head on, is this. How do we set thresholds for this digital data that genuinely reflect what matters to a patient?
Yeah, not just something that looks statistically significant on a chart, but actual meaningful improvement.
Precisely. Avoiding the statistical noise and finding the true signal of patient benefit.
Because these digital health technologies, DHTs, they're popping up everywhere in clinical trials now, aren't they?
They really are. And they're evolving fast. They're moving beyond just, passive monitoring. They're actually becoming new ways to measure health outcomes, almost like discovering new biomarkers in your daily life data.
Interesting.
And this is especially critical under the FDA's drug development tool qualification pathway. That's the FDA's formal process to say, yes, this digital measurement is reliable, it's fit for purpose.
Fit for purpose. So it can actually be used to help evaluate new drugs.
Exactly, which can potentially speed up approvals, get needed medicines to patients faster. That's the hope.
And these DEs, these digital endpoints, they're quite different from the traditional ways we measure outcomes, right? The COAs?
Oh, fundamentally different. Think about traditional clinical outcome assessments, COAs. They're often like questionnaires. Maybe you fill one out at a clinic visit every month, maybe less. It's a snapshot.
Right, a single point in time.
Yeah. But digital endpoints, they provide this continuous passive stream of data, like your watch tracking sleep quality every single night or activity levels throughout the day.
Okay, so it's constant, high-resolution data.
Exactly. And that presents, well, immense opportunities, but also some really unique challenges compared to those single data points from traditional COAs.
So revolutionary potential, you could say. But like any revolution, there are bumps in the road.
Significant hurdles, yeah. And the McCarthy paper really dissects these core dilemmas. Like, imagine you design a drug and statistically it looks like it works on paper. The numbers change.
Okay.
But then you ask the patient, how do you feel? And they say, honestly, I don't feel any different.
That's the gap.
That's the gap. And that highlights the first, and you could argue, the most crucial challenge. How do we make absolutely sure our digital measurements capture what actually matters to the patient experience, not just what a sensor can detect?
It sounds obvious, doesn't it? Focus on what matters to the patient.
Yeah.
But practically, how do you connect that subjective feeling, that experience, with these, objective streams of numbers from a wearable? What methods are actually working?
Well, that leads straight into the second big challenge they point out. Anchor selection.
Anchors.
Yeah, anchors. These are the established, validated measurements we use to compare our new digital data against. Think of things like a patient's overall impression of their health, a patient global impression, or PGI, or maybe a clinician's assessment, a CGI.
Okay, like a benchmark.
Kind of. With traditional methods, you compare maybe a before snapshot to an after snapshot using that anchor. But with continuous digital data, you've got this constant flow. How do you pick the right moment or the right summary statistic from that stream to compare it meaningfully to a single PGI or CGI score taken at one point in time?
I see the problem.
Yeah.
It's like comparing apples and oranges, or maybe apples, and a constantly flowing river of apple data.
Exactly.
Yeah.
It's like trying to match one specific wave in a constantly moving ocean to a single photograph. Which wave do you choose? How do you summarize the ocean's movement?
That makes sense. And then there's the individual variability, right? If I'm tracking my sleep, what feels like a meaningful improvement for me could be totally different for you, even if we had the same condition. Our baselines are just so different.
You've hit on the third major challenge, the data characteristics themselves. Determining meaningful change when you have such wildly different baseline values across patients, diverse populations in these long-term studies, it's incredibly complex.
So my normal sleep pattern versus yours.
Totally different, potentially. And your sleep might change day to day for reasons completely unrelated to the drug being tested. Stress, diet, who knows?
Right.
So we need to make sure the drug isn't just making your Fitbit think you're more active, for example, but that it's actually making you feel a real tangible difference.
Yeah.
It's separating that true signal from all the background noise, the statistical static.
And sometimes that static can look a lot like a signal, I imagine.
It absolutely can.
Okay, so we've got patient perspective, anchor selection, data variability. What else? Are there people challenges too?
Definitely. That's the fourth challenge the paper highlights. The multiplicity of stakeholders. You've got clinical teams, statisticians, patients themselves, regulators like the FDA, biostatisticians.
A lot of cooks in the kitchen.
A lot of different perspectives, and crucially, they often use different language, different frameworks when they talk about meaningful change. That can really slow things down, hinder progress when everyone isn't quite speaking the same language.
Wow, okay. These challenges sound pretty immense, especially, like you said, accounting for my unique daily fluctuations versus yours. So how do researchers even begin to untangle this? What's the new thinking, the methodological framework they're proposing to make sense of it all?
Well, what's really insightful in this paper is that they didn't reinvent the wheel entirely. They actually leveraged the FDA's existing patient-focused drug development framework, the PFDD guidance documents, specifically guidance 1 through 4 as their foundation.
Okay, so building on existing regulatory thinking.
Right. And they strongly advocate for blending qualitative and quantitative methods. So starting with the qualitative, actually talking to patients through interviews, focus groups, to find out what health aspects they prioritize, what really matters in their daily lives.
So listening first, not just deciding from the outside what's important to measure. That feels like a pretty significant shift for drug development, doesn't it?
It really is. Then, once you understand what matters, you collect the anchor data, like those patient global impressions, the PGIs we mentioned. You use anchor-based statistical methods to estimate meaningful change, but you also run sensitivity analyses to make sure those anchors are reliable and truly reflecting change.
Okay, so patient input, then robust anchor analysis.
And they don't stop there. They also bring in distribution-based metrics, things like looking at the standard deviation of the data or the minimal detectable change ways to statistically quantify what's likely just random noise versus what might be a real signal from the treatment.
Got it. Using stats to filter the noise.
Correct. And crucially, they suggest using tailored responder analyses and longitudinal modeling. This is especially important for those continuous digital metrics, trapping change within individuals over time.
Okay, that sounds sophisticated.
And they also propose something relatively new, a composite evidence approach.
Composite evidence?
Yeah, drawing conclusions not just from the statistical analysis and the qualitative patient input, but also from what they call injection-based approaches.
Injection-based, what does that mean?
It basically means you intentionally introduce a known change or stimulus, not necessarily a literal injection, but some controlled change, and you observe how the digital endpoint responds. It helps to validate how sensitive and accurate the digital measure actually is to real changes.
Oh, okay, like testing the responsiveness of the sensor or the metric itself.
Precisely. So it's a multi-pronged approach. Patient voice, anchor stats, distribution stats, longitudinal tracking, and this injection-based validation.
That sounds like a much more thorough way to go about it. So when they applied these kinds of methods, what did they find? What are the core takeaways for how we should be figuring out these meaningful change thresholds?
Well, the core revelation, perhaps unsurprisingly, is that our old ways of measuring health, those one-off clinic visits, those snapshots, they just don't hold up well against this constant flood of digital data.
And kind of break down.
Yeah, they really do. Imagine trying to capture the essence of that flowing river we talked about with just a single bucket sample. That's the kind of disconnect they found between traditional snapshot methods and continuous wearable data.
Right.
Think about it. An anchor measure like a PGI might be assessed at week 24 of a trial. but you're getting daily, maybe even hourly fluctuations in gait speed or sleep patterns from a wearable. There's often a fundamental misalignment in timing and granularity.
So it's not just that my baseline is different from yours, but even my own baseline is bouncing around day to day.
Yeah.
How does the paper suggest we deal with that inherent noise within just one person's data?
That's exactly where their most groundbreaking finding really comes in. Meaningful change isn't some universal fixed number.
It's not.
No, it's deeply personal. It shifts from person to person. And it can even change for the same person as their disease progresses or their life circumstances change.
Wow.
This really demands a radical rethinking of what improvement truly means in this new era of personalized health data.
So no single magic number for improvement.
Nope. They really stress the importance of within-patient measures. It's like trying to judge a runner's improvement fairly if they started on a different track each time, or even ran on different days with totally different weather. It doesn't make sense.
Right. You need to compare them to their own previous performance under similar conditions.
Exactly. The paper emphasizes understanding your personal baseline, your normal day-to-day or week-to-week fluctuations. That way, we're measuring your progress against your own unique standard, not just lumping you into a broad group average.
That makes intuitive sense.
And interestingly, they also found that anchors tied directly to patient perception, like that PGI, the patient global impression, are generally much preferred over clinician-based anchors like the CGI when you're trying to truly capture what matters to the patient from these digital measures.
That also feels right. The patient's lived experience should be the gold standard, shouldn't it? But historically, clinical trials have relied so heavily on what the clinician observes or reports. What are the biggest shifts needed, conceptually or methodologically, to really put the patient's view front and center with these digital tools?
That's a great question, and the recommendations are quite clear on this. First, they say you absolutely need to establish a precise conceptual framework right at the beginning.
Okay, lay the groundwork.
Yeah. Define the specific context of use for the digital tool. Clearly define the concept of interest. What exactly are you trying to measure? And be clear whether the endpoint is functioning as a clinical outcome assessment, measuring how a patient feels, functions, or survives, or more like a biomarker, measuring a physiological process.
So clarity upfront is key.
Absolutely. Second, ensure the whole process is patient-centered by grounding your anchor development in that qualitative patient data we discussed. Really understand what aspects of their condition impact them. And use patient-reported anchors like PGI whenever it's feasible and appropriate.
Okay, framework first, patient-centered anchor second. What about actually calculating the thresholds, the numbers themselves?
Right. For deriving the actual meaningful change thresholds, they recommend a robust hybrid approach.
Hybrid, meaning mixing methods.
Exactly. Combining those anchor-based estimates, the distribution-based metrics, and incorporating inputs from the qualitative work and those injection-based studies we mentioned. And always run thorough robustness checks, sensitivity analyses to make sure your threshold isn't overly dependent on one specific assumption or method.
Okay, so multiple lines of evidence feeding into the final number.
Precisely. And for continuous threshold methods dealing with that constant data stream, they suggest moving beyond just comparing group averages, like Group A improved by X amount.
Which is the traditional way, right?
It is. Instead, they propose focusing on individual level analyses, defining responders based on changes relative to their own baseline.
Okay, this individual level analysis sounds really powerful. Could you give us a quick, concrete example? How would that look different from a standard group level result?
Sure. Let's look with the walking speed example for a condition. A traditional trial might report. On average, patients on the new drug increased their walking speed by 0.1 meters per second compared to placebo.
Okay, standard finding.
Right. But what does that 0.1 meters mean for you? If your baseline speed was already quite high, maybe that's negligible noise. If you were severely impaired, maybe that 0.1 meters is life-changing. It means you can cross the street safely now.
Ah, I see. The average hides the individual impact.
Exactly. With individual level analysis, the focus shifts. We might define a responder as, say, a patient whose daily average stride speed decreases less than X meters per second below their own personal mean over a week, or increases by Y amount consistently. It's about measuring change that's significant at your level, against your baseline and variability.
That really does feel like a step towards truly personalized medicine, measuring what matters to me.
It is. It's a fundamental shift.
So, zooming out a bit, what about the bigger picture? Policy, regulation.
Yeah.
How does all this fit with agencies like the FDA or EMA? Does this paper address that?
It does raise that important question. The paper strongly emphasizes the critical need for ongoing policy discussions and stakeholder dialogue. Promoting harmonization across different groups, researchers, companies, patients, regulators is key.
So everyone needs to get on the same page.
Yes, including through those formal drug development tool qualification pathways with the FDA and EMA. The good news is the FDA has already started accepting DEs as passive monitoring COAs within their DDT program.
Okay, so progress is being made.
Progress is being made, but the authors stress that we can't just take the old methods used for traditional COAs and apply them directly to DEs. The existing methods must be adapted, perhaps significantly, to account for the unique characteristics of digital endpoints, the continuous nature, the variability, all the things we've discussed.
So it's not just a copy-paste job for the rulebook.
Definitely not. It requires genuinely new thinking at the regulatory level, not just adapting procedures in the lab or clinic.
Makes sense. It's fundamentally different data.
Precisely. And the paper really ends with a call for urgent public-private collaboration. We need industry, academia, patient groups, and regulators working together to develop standard methods and best practices to really expedite the validation and qualification of these incredibly powerful new tools.
Okay, so let's recap this deep dive into the McCarthy paper on digital endpoints and meaningful change. The fundamental takeaway seems crystal clear. This continuous passive data from wearables and sensors. It offers amazing sensitivity for measuring health.
Unprecedented sensitivity.
But leveraging it effectively demands totally new ways of thinking about what meaningful change actually is. The core revelation, as you put it, is that our old snapshot methods, those clinic assessments, they just kind of fall apart when faced with this constant stream of data.
They really do struggle. And the researchers identified several key pillars for making digital endpoints work effectively going forward.
Right, those pillars were.
Patient co-designed anchors start with the patient. Individual level thresholding focusing on changes that are relevant to you specifically. Using composite evidence, bringing together stats, qualitative insights, and those injection-based validations for a robust picture.
Okay, patient-centric, individual-focused, multifaceted evidence.
And critically, sustained regulatory engagement. Working closely with agencies like the FDA and EMA to validate these new methods and get them accepted.
And connecting this to the bigger picture, you mentioned this is the third paper in a series.
That's right.
So it really deepens the toolbox, the methods we have, and it highlights this urgent need for everyone involved, public sector, private sector, to collaborate, develop standards, and speed up the qualification process for these tools.
Absolutely. Because ultimately, this isn't just an academic exercise, right? It's about finding ways to get better, more effective medications to patients faster, measured in ways that truly reflect an impact on their actual daily lives.
Which brings us to a final thought for you, the listener, to maybe chew on.
Yeah, this idea that meaningful change isn't some fixed number, it's personal. It changes person to person, maybe even day to day for you.
So the question is, if future drug development can actually pinpoint what constitutes a truly meaningful change at your individual level based on continuous, personalized data flowing from your life, how might that fundamentally shift how we all understand health, disease, and even just the basic concept of feeling better?
What does it mean for you personally if the success of your treatment could be measured not just by a checkup every few months, but by daily relevant insights that are completely unique to your experience, your body, your life?
It's a pretty fascinating and maybe even hopeful future to imagine.